You are tuned to Iri-e-learning and I am your host, Teacher. In this States of Matter lesson, we will examine the evidence of osmosis. Check the description box to find the link to all the lessons for this unit. By the end of this lesson, we should be able to observe the changes that occur during osmosis and explain the process of osmosis in plant cells. Hey, it's experiment time! And the aim of our experiment is to observe the process of osmosis in potato cells. For this experiment, we will need the following materials: half of a teaspoon of table salt, which is sodium chloride, a ruler for measurement, a dinner knife for carving, a sharp knife to cut the potato, and an Irish potato. In some jurisdictions, the Irish potato is called the English potato or yellow-skinned potato. Let us take a look at the procedure for this experiment. Step 1. Cut the 4 centimeter thick slab from the Irish potato. Step 2. Use the dinner knife to carve a cavity in the center of the potato slab without puncturing the base. Step 3. Use blotting paper such as paper towel to remove any fluid released by the damaged cells during carving. Step 4. Place half of a teaspoon of table salt into the cavity of the potato. This experimental procedure is relatively simple, and if you decide to try this at home, make sure to have parental consent and supervision before doing so. When experimenting, it is essential to record all observations. This is when we use our 5 senses to gather information about what is happening during the experiment. Note that we avoid using our sense of smell, touch, and taste unless your teacher tells you that it is safe. So we rely mainly on our sense of sight and hearing for most observations. For this experiment, we can start by describing the physical appearance of the salt sample used. We can note the physical features such as the state of matter, whether it's a solid, liquid, or gas, the color, texture, and any other property that we can see. So the observable physical properties would include, first of all, we can see it's white. So the color is white and that it's in the solid state. And of course, it has a crystalline appearance. Therefore, we can say that the salt is a white crystalline solid at room temperature. Next, we should describe the changes that occur at specific intervals during the experiment. And for this experiment, we will record our observations in 40-minute intervals over 200 minutes. Also, we will only focus on the changes that occur within the cavity of the potato. So at 40 minutes, the salt crystals have a translucent and damp appearance. At 80 minutes, water is visible around the edges of the salt sample. Some of the salt crystals have dissolved. At 120 minutes, water accumulates in the potato cavity covering the salt sample, and the amount of salt in the cavity has been reduced. At 160 minutes, the water volume in the cavity has increased significantly. There is a further reduction in the quantity of salt crystals. At 200 minutes, a colorless solution that has a salty taste fills the potato cavity, and only a few salt crystals are visible at the bottom of the cavity. Now we're going to take a look at a sample observation write-up. Note that the observation report is typically written in the past tense because the write-up occurs after observing. However, this practice may vary depending on where you live. So make sure to follow the rules that apply to your school. So here we go. At the start of the experiment, a dry sample of white crystalline solid was placed in the potato cavity. Within 40 minutes, the salt crystals had a translucent and damp appearance. At about 80 minutes, water accumulated around the edge of the salt sample and some of the salt crystals had dissolved. At 120 minutes, more water accumulated in the potato cavity, submerging the salt crystals. Also, a notable quantity of the salt crystals had disappeared from the edges of the sample. These changes intensified over the next 40 minutes, observed by an increase in water volume in the cavity and a reduction in the quantity of salt crystals. By the end of the experiment, 200 minutes later, a colorless solution with a salty taste filled the potato cavity. Also, only a few salt crystals were visible at the bottom of the cavity. Now, take note of the disclaimer. Do not reproduce this document for official class assignments. The sample observation should only be used as a guide to assist you in writing a lab report of your own. After recording your observations, the next step is the analysis and interpretation, also called the discussion. At this stage, we explain our results and any significant actions taken during the procedure. Here we have a diagram illustrating the potato slab with the cavity filled with the salt particles, and salt particles are also solute particles. Now, before moving on, it is essential for us to keep in mind that potato is a plant, which means it's a living thing. Therefore, it is made up of cells. And cells, of course, consist of organelles embedded into a fluid medium called the cytoplasm, which is all enclosed by the cell membrane and the cell wall. So back to our illustration of the potato slab with the salt-filled cavity. You may have noticed that there are a lot of blue dots inside the potato compared to what we are seeing within the cavity. So if we take a look at the key at the top of the screen, you'll notice that the blue dots represent water molecules or water particles. The red represents the salt particles, which are solute particles, and the burgundy dots represent any solute particle that might be present within the potato cells. Now, since the blue dots represent water molecules, we can immediately see that there are more water molecules inside the potato than within the cavity. Therefore, the area of low water concentration occurs in the salt-filled cavity. Even though it's high in solute concentration, it's low in water concentration. Next, we can see that the area of higher water concentration occurs in the cells of the potato. Now, this difference in the concentration of the water molecules that's within the potato and in the cavity will generate what is called the concentration gradient. And once we have a concentration gradient, it will initiate the movement of water molecules from the region of high to low water molecule concentration. However, the water molecules must first pass through the cell membrane of the potato cells, which actually functions as the semi-permeable membrane. Therefore, we can say that the concentration gradient results in the net movement of water molecules from the cells, which is the region of high solvent concentration, through the semi-permeable cell membrane into the potato's cavity, which is the area of low solvent concentration. And this process is called osmosis. We will present 7 questions which you can use as a guide for writing a good discussion for this experiment. In fact, we have answered most of them, if not all, already. So question 1. Why was the cavity of the potato dried before adding the salt? 2. What structure functioned as the semi-permeable membrane in this experiment? 3. Where were the regions of high and low water molecule concentration? 4. State the direction of the net movement of water molecules. 5. What was the process demonstrated in this experiment? Give a definition. 6. Why did the salt crystals have a translucent appearance after the first 40 minutes? 7. Why did the salt crystals reduce in quantity as the water accumulated in the potato's cavity? So here is a sample discussion. During the carving procedure, some of the potato cells became damaged and seeped fluid into the cavity. Hence, the reason for blot drying the cavity. This step was essential for removing any trace of fluids that could interfere with the experiment results. In this experimental setup, there were specific conditions that prompted the movement of water. These included the areas of higher water molecule concentration within the cells and lower water molecule concentration in the salt-filled cavity. The notable difference in water molecule concentration between the two areas established a concentration gradient. The concentration gradient then initiated the movement of water molecules out of the potato cells, which is the area of higher water molecule concentration, passing through the semi-permeable cell membranes and into the salt-filled cavity, which is the site of lower water molecule concentration. This net movement of water molecules through the semi-permeable membrane from the region of high to low water concentration is called osmosis. As the water entered the cavity, the salt became moistened, which gave the salt crystals the initial translucent and damp appearance. As the water volume increased, the salt dissolved, producing the colorless salt solution, which had a salty taste and the reason why the salt crystals disappeared over time. Once again, take note of the disclaimer. Do not reproduce this document for official class assignments. The sample discussion should only be used as a guide to assist you in writing a lab report of your own. Let us review the lesson summary. A concentrated gradient develops between adjoining areas having different particle concentrations. The cell membrane of a plant cell functions as a semi-permeable membrane. Water moves by osmosis through the cell membrane of plant cells. An area of high solvent particle concentration is a dilute solution. An area of low solvent particle concentration is a concentrated solution. In plant cells, osmosis involves the movement of water molecules through the semi-permeable cell membrane from a region of high water molecule concentration, that's usually the dilute solution, to a region of low water molecule concentration, and that describes the concentrated solution. We have reached the end of our lesson. If you found this video helpful, remember to like, share, and subscribe. When you subscribe, you will receive notifications as new videos are uploaded. So until next time, I am Teacher with Iri-e-learning.